8 chapters have been completed in the first PUC and all 16 chapters completed in the second PUC. So the 9th chapter in first PUC biology is biomolecules, which is very, very important. So there are two types of biomolecules, that is macromolecules and micromolecules. When the biomolecules are the tissues of the plants and animals, when they are made to react with the trichloroacetic acid, two major groups are formed. One is acid soluble, another one is acid insoluble. Acid insoluble are the macromolecules like the proteins, carbohydrates, lipids, etc. Macromolecules, and their molecular weight is more than 800 daltons. Say for example, the macromolecules here, the water, it contributes for about 70 to 90%, and proteins 10 to 15%, and the nucleic acid 5 to 7%, carbohydrates 3%, lipids 2%, and ions 1%. So these are some of the macromolecules, but there are which are acid insoluble, and the acid soluble are many macromolecules. So actually the composition in the cell is hydrogen about 0.5 G, and then the percentage of the carbon is 18.5, and oxygen 65%, nitrogen 3.3%, and the sulfur 0.3%, and sodium 0.2%, and 1.5% of the calcium, and the 0.1 is the magnesium. The most abundant, hence the most abundant metal present in the human body is the sodium, but not the calcium. That is also very important. Say for example, if the tissues are heated, they lose water. Hence now such tissues have dry weight. Further, when they are heated, loses the carbon dioxide and water. Now they are called ash. This ash consists of inorganic substances like the sodium, potassium, calcium, magnesium, water, and compounds such as NaCl, calcium phosphate, phosphates, sulfates, etc. So that is about the ash and all. Coming to the proteins. Proteins are the organic compounds composed of amino acids. They contain carbon, hydrogen, oxygen, nitrogen. Few proteins contain sulfur and phosphorus also. Sulfur containing proteins are cysteine and methionine. This is also very important for your reading. Cysteine and methionine. Phosphorus containing protein is casein. Casein, the milk protein. And the proteins composed of chiefly amino acids. Amino acids are of two types. Our body cells are not capable of synthesizing certain amino acids. Hence they are called essential amino acids. There are about 10 essential amino acids like valine, lysine, leucine, isoleucine, tryptophan, threonine, methionine, etc. Or there are about 10 such essential amino acids. Our body cells are capable of synthesizing certain amino acids. They are called non-essential amino acids like the alanine, and then glycine, glutamine, glutamic acid, cysteine, tyrosine. Like this, there are certain amino acids. Body cells are capable of synthesizing. They are called non-essential. So this classification is also very important. And then there are aromatic amino acids like the tyrosine, phenylalanine, tryptophan are the aromatic. They have benzene ring structures. There are acidic amino acids too, aspartic acid and glutamic acid. And amino acidic basic amino acids glutamine, aspargine, and even more like lysine, arginine are the basic amino acids. So this kind of classification of amino acids is also important for your NEET examination. And proteins are the macromolecules, as I told you earlier. They are the heteropolymers, not the homopolymers. So the different type of proteins, for example, collagen is an intracellular ground substance, intercellular ground substance present, and antibodies which fights against, say for example, globulin. There are certain receptor proteins like sensory, that is sensory smell, taste, etc. Trypsin is enzyme, insulin is a hormone, whereas the GLUT, that is glutathione 4, which helps in the glucose transport. And coming to the structure of a protein can be studied from different views: the primary structure, secondary structure, tertiary structure, quaternary structure. Primary structure is based on the sequence of arrangement of amino acids. Any change in the sequence that alters the function of a protein, for example, sickle cell anemia, SCA. If it contains the beta 6th position chromosome, hemoglobin, if it contains the valine in the place of glutamine, that hemoglobin becomes abnormal and it renders the patient anemic. That is in sickle cell anemia, and it is also because of point mutation, where the GUG is present in the place of GAG. This is because of CAC and the DNA in the place of CTC. That is this is known as a point mutation, and I explained about this in the second PUC 5th chapter about sickle cell anemia. Anyhow, the primary structure of a protein based on the sequence of arrangement of amino acids. Any change in the sequence that alters the function of a protein. Secondary structure is based on how the amino acids are coiled into specific polypeptide chains. And three-dimensional structure given by the tertiary structure, in which we studied three-dimensional structure. It is the relationship between the primary and secondary structure. And according to the quaternary structure of a protein, it is determined first in hemoglobin. It consists of two chains, two alpha chains and two beta chains, 4 chains altogether, and 574 amino acids are present. So this is about the structure of a protein. At isoelectric pH, the carboxyl group as well as the amino group get ionized. They are called Zwitter ions. That is also very important as far as the protein is concerned. You will study the structure of protein in even the biochemistry in detail, giving also the structures. The next to this, the structures of some simple amino acids are given. Say for example, glycine. And if the R group is only H, it is glycine. R group is CH3 alanine. If the R group is CH2OH, that is serine. And these structures are given, and these things are very important. Also, all the structures you are expected to study in the textbook given, that is important for your NEET examination. If both carboxyl group and the amino group attach to same carbon atom, they are called alpha amino acids. They are called alpha amino acids. If the methyl group, that is the C group, that is attached to this, there are many things called, many groups called R group. R group differs from one amino acid to another amino acid. So that is about the proteins. And the carboxyl group of one amino acid binds with the amino group of another amino acid, forming a bond called peptide bond. So there are 3 amino acids in dipeptide, but amino acid residues are 3. It is often confusion. It creates a confusion. How many bonds are present in peptide bonds are present in dipeptide means single peptide bond is present. So it is a confusion, creating a question usually that will be asked. So this is about the proteins which are the macromolecules, and the peptide bonds are present. And the linkage is CONH linkage called amide linkage can be seen in the proteins. Coming to the lipids, lipids are the esters of fatty acids and glycerol, and structure of and energy in organisms stored in the form of triglycerides, that is CH2COO-R1, CHCOO-R2, CH2COO-R3. So it is a compound triglyceride where the R group differs. And the number of carbon in the lipids exist in two forms, oils and fats. Oils have low melting point. For example, gingerly oil will have low melting point. This is also very important. And based on their saturated fatty acids and unsaturated fatty acids, and arachidonic acid. So that is consists of 20 carbon atoms, and then 20 carbon. And then palmitic acid, which contains only 16, and the oils have low melting point. For example, the gingerly oil, and simple lipids present in the cell membrane. As I told you earlier in the previous chapter, the plasma membrane consists of these lipids. These lipids, especially the lecithin, structure of lecithin also very important. It is CH2COO-R, and then CHCOO-R, and then CH2 not COO. Here it is phosphate group attached to phosphate group CH2, and attached to last CH2 N, and then attached to this there are 3 methyl groups, that is CH3, CH3. So this is the structure of lecithin. This is also very important. And the simple lipids, the compound lipids, and the derived lipids like cholesterol structure is also given. Parahydrophenanthrin ring structure, it has 2 benzene rings and 1 more benzene ring above this, and then heterocyclic, then 5 angled, and then that is the structure of cholesterol also very important as far as the lipids concerned. So proteins, lipids, coming to the carbohydrates. Carbohydrates are broadly classified into sugars and non-sugars. Sugars are sweet to taste. They are soluble in water. And then the sugars are soluble in water. Sugars are of 2 types, the monosaccharides and oligosaccharides. Monosaccharides contain single sugar in it. Number of sugar units 2 to 10 in oligosaccharides. That you will study in detail in the biochemistry. That is in chemistry, you will study about the monosaccharides. Again, aldoses and ketoses, triose, and then tetrose, pentose, hexose. Pentose aldose sugar present in nucleic acids is ribose. That is very important for your NEET examination. And hexose aldose sugars are glucose, galactose, mannose, etc. And hexose ketose sugar is the fructose, and disaccharides are sucrose, maltose, and then lactose. These things are very important for the biochemistry as well as structures of some not the carbohydrates. Here the sugars given, that is the ribose sugar and the deoxyribose sugar. Ribose sugar structure and deoxyribose sugar, it is C5H10O5 ribose sugar present in RNA, deoxyribose sugar present in the DNA, that is C5H10O4. The second carbon lacks the oxygen molecule, the structures given in the textbook, so ready for that. And then next, the polysaccharides given. So it is the polysaccharides, very important. The polysaccharides. Polysaccharides are the non-sugars, usually insoluble in water, and the structural polysaccharide in plants is the cellulose, and the structural polysaccharide in some arthropods it is chitin. exoskeleton, for example, of cockroach consists of this chitin, and the inulin is a polysaccharide composed of fructose. So storage polysaccharide in plants, that is the starch exists in two forms, amylose and amylopectin, and storage polysaccharide in animal is glycogen. Why so far not asked, this is also important, why starch takes up the iodine immediately but not the glycogen? So starch is a storage polysaccharide in plants, and in animals it is the glycogen. But why takes up starch takes up iodine immediately? It is because of highly coiled structure, that is the structure of the amylopectin and amylopectin. It is highly coiled, hence it takes up the iodine but not the glycogen. That is also given in the book. And there are certain heteropolysaccharides. They contain other than the carbohydrates. For example, NAGA, N-acetyl galactosamine. N-acetyl galactosamine or peptidoglycan, which is present in the cell wall, which is heteropolysaccharide. Another heteropolysaccharide given is the chitin. Chitin is another heteropolysaccharide that is given in textbook, that is present in the arthropods. So that is about the carbohydrates, proteins, lipids. Coming to the nucleic acids, as we know very well, in the carbohydrates, the sugars are joined by the glycosidic bonds are present in carbohydrates. Peptide bonds are present in proteins. Ester bonds are present. In nucleic acids. There are two types of nucleic acids, DNA and RNA. I explained in detail about this. What given in the biomolecules is DNA is a double stranded, RNA single stranded, deoxyribose sugar structure given, and ribose sugar here it is the ribose sugar, and nitrogen bases present in DNA are AGCT, AG are called purines, adenine guanine, cytosine thymine called pyrimidines, and structures given, that is very important in this chapter. Adenine is a 6-amino purine, and the 6th group it is the amino group. 2-amino 6-oxy purine is guanine. 2-amino 6-oxy purine. And purines are larger than the pyrimidines. And that is about adenine and guanine. They are the common purines for both DNA as well as RNA. So the pyrimidines, cytosine, and it is 2-oxy-4-amino pyrimidine, that is the cytosine. 2,4-deoxypyrimidine is uracil, and 2,4-deoxy-5-methyl pyrimidine is thiamine. And the phosphorus present in the nucleic acid is H3PO4, that is orthophosphoric acid. If it binds with the 5th carbon atom of the sugar, that is 5' end, and if it is binds with the 3 carbon atom of the sugar, that is 3' end. Hence, DNA is a double helix consisting of 2 strands which are arranged spirally and antiparallelly. Body are the framework composed of phosphates and sugars, and steps in between them, rungs in between them composed of nitrogen base pairs. The base pairs of DNA are at GC. Adenine binds with thymine with 2 hydrogen bonds or vice versa. Guanine binds with cytosine with 3 hydrogen bonds or vice versa. This kind of binding is very specific. And DNA strands takes a turn of 36 degrees. That is very important. 36 degrees. And the pitch of DNA is 34 angstroms, and the internucleotide space is about 3.4 angstroms. That is also very important. And there are anabolic processes and catabolic processes, and apart from this, secondary metabolites are present, which are very important. The secondary metabolites are the toxins like abrin, the present in Abrus picatorius, and resin, and essential oils like the lemongrass oil, and alkaloids like morphine, codeine, etc. And then the pigments like anthocyanin and carotenoids. very important, and lectins like concanavalin are the lectins, and drugs like curcumin and vinblastine. Curcumin and vinblastine are the drugs. These are the secondary metabolites. And coming to the last aspect here in this chapter are the enzymes. Enzymes are the biological catalyst enzymes. They are the catalytical proteins. They are composed of only the proteins, catalytical proteins. Enzyme is a whole called holoenzyme, the protein part called apoenzyme, the non-protein called the cofactors. Cofactors are of three types of prosthetic group, coenzymes, and metal ion group. This is the non-protein part called the cofactors. Coenzymes are the cofactors which are loosely bound to the protein part, that is the enzymes. Example, NAD and FAD, nicotinamide adenine dinucleotide, where it contains vitamin niacin, the B4 vitamin, which is very important, NAD, and FAD, flavian adenine dinucleotide, which contains B2 vitamin, that is the riboflavin. So this is about the coenzymes. And coming to the prosthetic group, catalases and peroxidases are the examples for the prosthetic group given. And peroxidase, which oxidizes the 2H2O2 into 2H2O2, 2H2O2, that is the hydrogen peroxide into the water and the oxygen, that is peroxidase. And catalase plays an important role in synthesis of the chlorophyll, and the metal containing the cofactor, that is the zinc present in carboxypeptidase. So this is about the enzymes, this one. So all the enzymes, they end with suffix ASE. They end with the suffix ASE. And the different types of enzymes also given, that is transferases, which plays important role in the glycolysis, transferases, and the oxidoreductases like lactic acid dehydrogenase enzyme, and the hydrolases like the digestion, which enzymes which we study in digestion, like trypsin, chymotrypsin, all those things. Isomerases, glucose 6-phosphate is converted into fructose 6-phosphate during glycolysis in the presence of hexoisomerase. Isomerases, liases, which bind, ligases, they bind, and liases play an important role in, that is in Krebs cycle, binding, the water molecules where comes in these reactions. Thus, the different types of the enzymes are transferases, oxidoreductases, hydrolases, isomerases, liases, ligases, and different things given in the book. That is also very important. And then the enzymes, they act as the catalyst in any reaction. They act as catalyst. So for example, in glycolysis there are about 10 steps, and each step is catalyzed by specific enzymes, and C6H12O6 gives 2 C3H4O3, that is 2 molecules of pyruvic acid, and 2 H2, that is the products formed during the glycolysis. And if enzymes are present, the reaction will be very fast. Before that, how the enzymes work means it has an active site, and the substrates will have binding site, and this active site sometimes shapes the binding site of substrate, then fit into the each other, or straight away binds in a lock and key mechanism. And the enzymes present in the active site breaks the substrate site and then binds with this one, and finally unaltered enzyme get releasing the different types of the products, and the unaltered enzyme is ready to bind with another substrate. This is how the enzymes work. And the enzymes, if enzymes are present, the reaction will be fast. So, for example, if carbonic anhydrase is not present, that is CO2 plus H2O gives H2CO3. That is, in human respiration you will study about when carbon dioxide dissolves in water, it gives carbonic acid H2CO3 in the presence of an enzyme carbonic anhydrase. If carbonic anhydrase enzyme not present, only 200 molecules of H2CO3 are formed per 1 hour. If enzyme is present, 6 lakhs produced in 1 second. In a second, 6 lakhs molecules. So you see how fast the reaction in the presence of catalyst takes place. And reactions 10 million times faster if the enzymes are present. That example is also given. And there are certain factors which affect the enzymal activity. That is, the number 1 is the temperature, and above 40 degree centigrade temperature the enzyme activity will be affected, and optimum range of temperature is required for the enzymal activity. At the low temperature enzymal activity inhibited, even at the high temperature only the optimum range of temperature, that is 37 degree in case of mammals, which is important. And it is the concentration of the substrate also plays an important role in the enzymal activity. If the concentration of substrate will be increased, then the rate of reaction also increases, but to a certain extent. Later that the active site of enzymes will not be available at that one point, that is Vmax, that is, it is the concentration of substrate at which the rate of reaction half of the maximum value, Michaelis Menten constant, that is given in detail in chemistry, that is half Vmax. Michaelis Menten constant is half Vmax. So that is also very important. The other factors, as we know, the pH and all those things, again in the chemistry it is given. And how they increase the rate of reaction means by increasing the activation energy. Activation energy means it is the extra energy required for the reactants upon collision to give a product called activation energy required for the reaction. That is very, very important. And the substrate and the product, you know, if S and P, if P is at the lower level than the substrate level, the reaction is to be exothermic. So a difference in the average energy content of a substrate from that of this transition state is called active energy, and that graph is given. So the activation, about activation energy and all those things, and you can study in detail even in chemistry also. And then there are certain inhibitors of enzyme action. These inhibitors, they have similar structure of substrate and they compete with the substrate. This is very important. Certain inhibitors, they compete with the substrate, and instead of substrate they bind with enzyme, and hence no substrate is available, hence the enzymal activity will be inhibited. So the best example given is malonate. Malonate inhibits succinic dehydrogenase. Instead of succinic dehydrogenase, the enzyme malonate inhibits the succinic dehydrogenase as it resembles the succinate in its structure. So malonate here, it competes with the succinate, and then it doesn't allow the succinate to bind with the succinic dehydrogenase, but this itself binds with the succinic dehydrogenase and inhibits the action of succinic dehydrogenase. Thus, the malonate inhibits succinic dehydrogenase as it resembles the structure of the succinate. That is very important. The only one thing I have forgotten here in this chapter is the lipids are the macromolecules, as they are acid insoluble. But the lipids can be obtained only are less than 800 Daltons, because the lipids are present in the cell membranes. So in order to obtain, we have to crush the lipids. Then the lipids size becomes less than 800 Daltons. And truly speaking, the lipids are not the macromolecules. It is because of this reason when we obtain less than 800 Daltons. But lipids are acid soluble, hence they are macromolecules. So, dear students, I completed even the 9th chapter, that is in the first PUC, and all the chapters in second PUC, line by line. Go through it, and important points here and there I stress, those things only appear for your NEET examination. It is very easy to crack the biology as well as the NEET examination, provided if you give sufficient time to read biology as it is the easiest when compared to the other subjects. And then the thing is, the textbook is only the textbook, and all the sentences in the textbook are very important. Hence, the biology textbook is Bhagavad Gita for your NEET examination. Okay, thank you. Please subscribe.